Today we're going to talk about adaptive evolution, and that means that today is going to be all about the different kinds of natural selection that there are. It's going to be about the vocabulary that evolutionary biologists use to describe selection. It's going to be about rates of evolution, why evolution is sometimes very fast, sometimes very slow, and it's going to be about the different contexts in which selection occurs. So we'll talk a little bit about sexual selection, we'll talk a little bit about group and species selection, things like that. All of these things that I mentioned today are going to be coming up again and again. So this is just part of the intellectual toolkit for dealing with the course. This is an outline of the lecture, and since it's a whole lecture, it's in pretty small type, and I don't expect you to read that off the board. But I do want you to have it so it makes it easy for you to review this when you download it and you look at it in your notes, because it does summarize the main points. Basically, what I'm going to do is tell you that evolution can be either adaptive, in which case it has been driven by and shaped by natural selection. It can be neutral, in which case it's been dominated by drift, or it can be maladaptive. So evolution does not only produce things that work well. Evolution produces things in which stuff can go wrong. And sometimes evolution just wanders around. Now, adaptive evolution is not about the survival of the fittest. That is a phrase invented by Herbert Spencer in the 19th century that has had a long shelf life, and it's wrong. Adaptive evolution is about design for reproductive success. It's all about how many children and grandchildren you have and whether you do it better than somebody else that's in the population. It's always relative. Natural selection is like the tale about the Buddhist monk and the disciple who were attacked by the tiger, and the disciple says to his master, "Oh, master, we're going to be killed because we cannot possibly outrun the tiger." And the master says, "No, I just have to run faster than you." Well, selection is always relative. Okay, it always depends on what the picture is of reproductive success in that population at the time that it's happening. Now, I'm then going to discuss when selection is strong and when it can be slow, and I will tell you something about the rates, the units in which evolutionary rates are measured, and then I'll run through types of selection. Now, there are going to be two questions that pose puzzles that come up in this lecture. One is going to be, what will happen if directional selection continues for a long time? Can that continue? And if it has to stop, then why should it stop? And the other question will be, how can we explain that even though evolution can really be extremely fast, that sometimes things don't change for hundreds of millions of years? So you have to be able to come up with enough intellectual tools to be able to handle that range of variation in evolutionary outcome. Both of those things really do happen. Nothing for a long period of time or incredibly fast. So here's incredibly fast. Antibiotic resistance. It is a curious and striking cultural fact that in the United States, when people talk about antibiotic resistance in television and in the newspapers, they almost never mention the word evolution. They say it emerges or it develops. But in fact, this is the poster child for rapid evolution. If you can see here, roughly the years in which antibiotic resistance evolved in these diseases. If we develop a new drug and release it in the UK in 2009, resistant strains of bacteria will have evolved and will be in hospitals in the UK within 6 months. And those resistant strains of bacteria will be observed in hospitals in Hong Kong within 2 years. The bacteria will have moved around the earth as people move around the earth. The drug industry is in a co-evolutionary arms race to try to keep up with the bacteria as they evolve resistance, and we have gradually been losing the arms race. So if you read in the newspapers about multiply resistant Staphylococcus aureus, which is MRSA, It is starting to crop up now out in the community. It's not just confined to hospital emergency wards and intensive care units anymore. It's starting to spread. And if Staph aureus picks up resistance to vancomycin, and which is one of our last lines of resistance against it, it's going to be very difficult for surgeons to do operations in the confidence that they can keep their patients from dying after they have surgery. So this is serious stuff. Most resistant bacteria live in hospitals because that's where most antibiotics are used. And the number of hospital-acquired infections is about 2 million per year in the United States, and it's estimated that about 90,000 people who did not have a bacterial infection when they went into the hospital, got in the hospital, and then died from that bacterial infection. And in fact, it looks like this is a serious underestimate because this is the official report, but if you look at what the hospitals ask for in terms of money from the insurance industry, it's about 10 times higher than that. Okay? So, by comparison, this is how many people are dying in 2005 from AIDS in the United States, influenza, and breast cancer. And you take that and you multiply it to the planet, and you can see that the evolution of antibiotic resistance is a pretty serious issue. The economic burden in the US 4 years ago was $80 billion a year. And this is a problem which is caused by strong directional natural selection eliciting a rapid evolutionary response. So in the next few slides, I'm going to be talking about what I mean by directional and what I mean by rapid. By rapid, in this case, if you have a normal-sized bacterial population in your body, and I give you an antibiotic, the probability is that if you don't do your antibiotic treatment correctly, within about a week or two, you will have resistant bacteria in your body. Finish your antibiotic treatments. Never stop in the middle, okay? Kill them all off. Here's a good example of rapid evolution in nature in a fish in ecological time. It's one of a series of cases that accumulated in the 1970s and 1980s that demonstrated that evolution isn't all about dinosaurs and millions of years and slow, steady change. Evolution in stuff like the color of the male's body, the number of babies the female has, how fast they grow, all kinds of ecologically and behaviorally important properties can happen real quick. This was done by doing an experiment in which the guppies interacted with predators. This is a cichlid fish, Crenocycla. This is the pike killifish, Rivulus, and it was done in Trinidad by David Resnick, who was at Riverside. Now, the setup in Trinidad basically is that there is a mountain range on the north end of the island, and there are lots of little streams that are going down the mountain range into a river, and they go over waterfalls. And the fact that the stream goes over a waterfall has prevented the large predators from getting up above the waterfall. And above some of the waterfalls, there were no fish at all. So what Resnick did was he took fish that had evolved for a long time with predators below the waterfalls, and he put them above the waterfalls. And he did replicates. It was a nice system. There were lots of streams. You could do it 4 or 5 times to make sure it was a consistent pattern. And these are the results. The life history traits, that means how big they are when they're born, how old and how large they are when they mature, how many babies they have, and how long they live, all evolved rapidly. So they responded quickly. The fastest rates of evolution were measured in things that occur early in life, so the number of babies in the first brood, how big the babies were in the first brood, how fast the babies grew. That all changed quickly. And basically the pattern was this. If the guppies are under a high predation regime, they mature earlier and they have more smaller offspring. They have a shorter life. This all has something to do with the evolution of aging and why we grow old and die. And they had more smaller offspring. Okay? The males were less colorful and they displayed more discreetly. guppy courtship is normally a fairly elaborate thing. The male, whom you can see is really brightly colored, also has an elaborate display behavior. And he will dance up in front of the female and he will wave his fins back and forth, and then he will dart in and try to mate. And the female prefers males who have bright orange spots. The bright orange spots probably were originally a direct indication that the male was good at catching crustaceans, because the crustaceans have carotenoids in them. So they catch amphipods and shrimp and things like that, and then reprocess the chemicals, and they can make orange with it. That was an indication that a male was a good forager, and so the female might select that male because then her babies also would be good at catching food. However, the male is dancing in front of the female, and that makes him a sitting duck for Crenocycla. And as we'll see a little bit later in the lecture, sexual selection involves a direct trade-off between mating success and survival. And these guys were displaying frantically to get mating success at the risk of being snapped up by a predator. And the ones that survived were the ones that simplified their display behavior. Okay? So this all happened pretty quick. Now, how do we measure it? Well, currently, and there's been a bit of controversy about this, but currently, the preferred unit of measurement is a Haldane. And a Haldane is a change in the mean value of the population by 1 standard deviation per generation. So I'm going to tell you what a standard deviation is, and I'm going to tell you who Haldane was. Haldane was the son of the Lord Admiral of the British Navy, who commanded the British Navy in World War I. And he was a brilliant polymath. He was fluent in Greek and Latin, as well as mathematics and biology. And he did foundational work in biochemistry and on the origin of life, as well as in population genetics. For many years, he was professor at University College London. He was a communist and a socialist and a social reformer who had a romance with the Soviet Union and then became bitterly disillusioned when he discovered what had gone on in the gulags in the 1950s. When he got intestinal cancer, he retired from his position in London, and he took a job in India, and he taught a whole generation of population geneticists in India before he died in 1962. A very interesting guy. And actually, there is a lot about the social impact of science that you can learn from reading about J.B.S. Haldane. The biography is just called J.B.S. Okay. This is a standard deviation. It is an empirical observation supported by an elementary theorem of mathematical statistics called the central limit theorem, that most population distributions look like a bell-shaped curve. It's called the normal distribution. It was formalized by Gauss. Sometimes it's called a Gaussian distribution. And the shape of the curve and its spread are basically measured by the standard deviation. So the mean value is at the center here, and the distribution is theoretically symmetrical. In practice, it's quasi-symmetrical. And the degree of spread is measured by the standard deviation units. Okay, so within 1 standard deviation, you will find on each side 34.1, or with both 68.2% of all of the individuals observed in the population. So 1 Haldane basically would take a population that, say, had a mean value, suppose it was for body size, maybe a body size of 10 grams, and if it had a standard deviation of 2 grams, and it was evolving at a rate of 1 Haldane, it would move that mean from here to here, 1 standard deviation unit up. And it would, the population mean in the next generation, instead of being 10 grams, would be 12 grams. So that's the meaning of the Haldane. Here are some measured Haldanes. Okay? So for those guppies in Trinidad that were evolving pretty fast, the number of spots in the area of orange spots, when you took away the predator, and suddenly being brightly colored wasn't risky anymore, and females liked it, so it was good to be brightly colored, those spot numbers increased quickly. They were increasing at about 0.7 Haldane. In the Galapagos finches that Peter and Rosemary Grant studied, they go through El Nino, and during El Nino, it's a strong selective event. So about every 10 years, there's a strong selective event on the Galapagos finches. And during El Nino, they were evolving at about 0.7 Haldane in body size. They're getting bigger. And then in the other years, they were getting smaller. So they fluctuate, okay? They go up and down depending upon the El Nino conditions in the Galapagos. There have also been lots of measurements of slower rates. For example, since the extinction of their competitors in the late 19th century, the surviving Hawaiian honeycreeper, the Iivi, has been evolving a shorter bill. And that's been a very slow rate of evolution. The migratory timing of Columbia River salmon has been changing as a result of the human fishery on them. All of the fished populations of the world are evolving under the pressure of human fishing. Most of the fish in the world are getting smaller. Many of the stocks are collapsing. It's producing a change in the time of year that the Columbia River salmon run up the Columbia. This is also due to the building of dams on the Columbia. So this is a human-induced selection pressure. These are fairly slow rates. So what does this mean if we just try to think about these rates and evolutionary time? A Galapagos finch is about 25 grams. They're about the size of a house sparrow. They evolved during El Nino at about half a gram a year. What if the El Nino conditions persisted forever? What if it wasn't the southern oscillation that was driving the rainfall pattern in the Galapagos? What if it just stayed warm and wet for a long time in the Galapagos? Well, that would produce directional selection, and if you did it for 100 years, it would turn a 25-gram finch into a 75-gram finch. Basically, it would take a finch and turn it into a small robin. Okay? If you did it for 10,000 years, it would turn it into a turkey. Now, finches as big as turkeys don't do very well in a finch habitat. They are living in a place where they hop around in bushes. They are living in an environment in which food is sometimes very hard to come by. I've been observing the turkeys that live near my garden in Hamden trying to get up into the trees next to Lake Whitney to pick the berries off as winter has come on and it's gotten very cold. They're pretty clumsy. So what will happen if you keep a strong directional selection going on finches? What would happen to humans if there were strong directional selection on humans to increase in body size? What would happen if we got turned from, say, 50 to 80 kilo primates into 3-ton primates? How long could that go on? One of the fastest rates of evolution ever measured in the fossil record was when elephants went onto islands in the Mediterranean and turned from 12-ton elephants down into things about the size of a St. Bernard. Okay, they did it in less than 100,000 years. They did it because they were food limited and they'd been released from predation pressure. Okay? So how far can that process go? These are quick changes that we're describing. The finches are moving pretty fast. The guppies are moving pretty fast. The elephants change pretty quickly. But if you look over the whole spread of evolutionary time, over hundreds of millions of years, things stay within a fairly narrow envelope of body sizes. Why does that happen? So if we look at microevolutionary rates, and by the way, there are good papers on this. If you're interested in rates, this is a good paper topic. Lots of measurements, lots of argument about why. They vary from very fast to very slow. The fastest are in the finches and in the Trinidad guppies. There have been lots of rates measured in Hawaiian mosquito fish and Hawaiian honeycreepers, so there are lots of estimates available. And interestingly, the shorter the period over which the rate is measured, the greater the maximum rate. So if you measure a rate by making comparisons between two populations that have been separated for hundreds of years or hundreds of generations, it's usually fairly slow. And if you focus in and you just look at a brief period, it can be very fast. Why do you think that might be? Why might we measure a faster rate when we do so over a shorter period of time? If we measure it over a short period of time, sometimes it's faster. If we measure it over a long period of time, it's slower. Does that suggest anything about what the pattern might look like that I'm about to draw on the board? Yeah. You got it. That's all it takes. It just has to go up and down. If I measure it over this period, it looks pretty fast. If I measure it over this period, it looks pretty slow. That's all it is. Okay. The take-home message from many studies done in the 70s, 80s, and 90s is that evolution can be very fast when populations are large and selection is strong. And the reason for that is that big populations have lots of genetic variation. So there's a potential for a big response to selection. Small populations don't have so much genetic variation. So even though selection might be strong, they can't respond so well. This point, the shorter the time interval over which you measure the rate, the higher the maximum rate. And here's one reason why you can't take Galapagos finches and turn them into turkeys and then turn the turkeys into ostriches and then turn the ostriches into moas and then have the moas turn into Tyrannosaurus rex. Okay? As you push things very far in any direction, there's an internal process that converts the directional selection into stabilizing selection. And those are the trade-offs, the linkages among traits. If you try to make a finch very large, then although it may be gaining something in terms of, say, food capturing ability, it is giving up maneuverability. If you try to take elephants and make them very small, then at some point they are not going to be able to compete with other elephants for food supply, even though there may not be any predators there. There are all kinds of biomechanical linkages within bodies where trade-offs are involved. So if you look within the organism, you see that it's a bundle of linkages and compromises. And every time you try to change one trait, you have a byproduct, you have an implicit selection going on other traits. So although you may be realizing a benefit in one, you are paying a cost in the others. The most striking example we've seen of it in the lecture so far is the guppy, the male guppy. If he evolves to be bright and a wonderful dancer so that females just love to mate with him, he will get killed by a predator. That is about as straightforward and brutal a trade-off as you can imagine, okay? But these go on all over the place, and some of them are very subtle. Now, why is it that sometimes traits evolve very fast and sometimes very slow? This is a picture of club moss, a Lycopodium. If I were to take you out into the woods of Connecticut in the springtime, you would see them all over the place. And if I were to put you in a time machine and take you back 400 million years, they wouldn't look any different. This is Latimeria. This is a coelacanth. If I were to put you into a research submarine off the Comoro Islands in the strait between South Africa and Madagascar, between Malawi and Madagascar, and we went down at night to a depth of 300 to 600 feet off the volcanic slope of the island, we would find these guys cruising around in midwater. They have spent the day in caves, and they come out at night into the midwaters of the Earth's oceans. And apparently they have been doing this now for going on 150 million years. They haven't changed at all. By the way, they have an egg the size of an orange. They're interesting. They're pretty effective predators too. They are ambush predators. They drift around and then they suck things into their mouths by a big kind of vacuum suction device. It's a common method of fish feeding. So they're living fossils. Now, why haven't they changed? Look at what's happened to their relatives. The club mosses had relatives at the time that looked about like them, that since then have turned into redwood trees, orchids, wheat fields, you name it. These guys still look the same. Latimeria had relatives that since then have turned into marlin and white sharks and reptiles, birds, mammals. It hasn't changed. So we have these two things to understand. We have to understand how evolution can go really fast, antibiotic resistance, guppies, finches, and why sometimes it is so slow. Any ideas on this one? Is this the first time you've hit this problem of why evolution is sometimes so slow? Yeah. Right. Okay, that's one kind of explanation, and I think it's certainly a plausible one. It's not the only one, but it's certainly one kind. So his argument is, the reason these guys haven't changed is that they're really good at always finding the same kind of environment so that they are never exposed to change. So if their environment moves around the globe, they track it. Now, remember, between 140 million years ago and now, the Earth went through a huge meteorite strike. The dinosaurs went extinct. Heavy stuff happened back there at the end of the Cretaceous, and Latimeria just cruised around, and it hasn't changed very much. Now, the argument is actually probably most convincing for marine invertebrates that make larvae that can go out and spread through the ocean for thousands of kilometers. And in fact, we know from the behavior of marine invertebrate larvae, so now I'm talking about worms, barnacles, clams, stuff like that they like to settle on places where there are successfully growing adults of their own species. They smell that out very carefully, and that's where they settle. So basically, the larvae are selecting the habitat in which the adults will be selected by natural selection. That means that they manage themselves to generate stabilizing selection over hundreds of millions of years. And arguably, Latimeria has done the same thing. It's been living in lava tubes on the sides of submarine volcanoes at 300 to 1,000 feet for a long time, and that habitat's always been around. Any idea for another explanation of stasis? That's an externalist explanation, okay? It relies on aspects of the habitat and the way natural selection is operating on the organism. Anybody got an idea for an internalist explanation? No?
There's genetic mechanisms for regulating DNA copying and improve as well as amplification.
I very much doubt that a lack of mutations was ever the reason that things didn't change. You've got 4.6 in you that are new since your mom and dad, for example. Yeah. Well, yeah, the problem with that over a long period of time, Greg, is that if it's really a small population, it's more likely to go extinct, and these things are out there for hundreds of millions of years. So that one's a little difficult. Other ideas? Well, there's a whole school of thought that says that this kind of thing is due to developmental constraints. That development has constrained the organisms. so that they couldn't evolve in certain ways. And that's plausible for certain major features of the body plan that are determined very early in development and involve developmental tissue relationships and things like that that are obviously hard to change. It's not so plausible for some of the smaller details of these creatures. So I think that the actual explanation is probably a mixture of these things. There probably is some phylogenetic or developmental constraint. Things that happened a long time ago in the way organisms were built are hard to change. And they've been constraining the things that can change more rapidly. But I think you'll find if you get into this that it's a huge and controversial literature on it. Okay. Kinds of selection. Now we go through another one of these vocabulary building exercises. And I'll try to illustrate a few of these, but I just want to get these words out there, and I want to get them into your minds so that you can start to think about the fact that natural selection comes in lots of different flavors. We can talk about directional, stabilizing, and disruptive selection, natural and sexual selection, frequency-dependent selection, and then selection acting on individuals, on kin, on groups, and on species. So each of these is cutting the selection cake in a different direction. But it is all of these different things. So, directional, stabilizing, and disruptive. Basically, what's going on with directional selection, that's making the Galapagos finch into a turkey, is that the fitness gradient is linear. That means that if the fitness of something over here is low and up here is high, that means that natural selection is selecting for, say, bigger things. If body size on the x-axis is going to the right. And it will take a distribution that looks like this, and it will move it to the right. So if this is one standard deviation, then this amount of movement is one Haldane, right here. Stabilizing selection is actually what we were just invoking to argue that the coelacanth didn't change. It was living in a habitat where it was always good to be like a coelacanth, and natural selection was selecting out things that didn't look like coelacanths, whether they were larger or smaller, or their fins were different shapes, or things like that. So they tended to stay the same. That means that we were selecting for the mean of the population, and we were discarding the extreme values. Who in the room is under 5'5", and also who in the room is over 6'1"? Raise your hands, please. Okay, if there's stabilizing selection on human height, you guys have no grandchildren. Can I see the hands of everybody else? Hey, you made it. Okay? That's stabilizing selection. It means selection for the mean value and a selection against the extremes. Be happy that that doesn't appear to be the only thing going on in humans. Disruptive selection is selection against the mean and for the extremes. And it will take a bell-shaped curve like this, it will knock out the mean value, and in the next generation, it will push it apart like that. Okay? So if we look for examples, strong directional selection will produce very rapid evolution. We saw that with antibiotic resistance and the guppies. It can continue. It usually gets converted into stabilizing selection. Disruptive selection causes, historically, things like the conversion of similar-looking gametes into quite different gametes. So disruptive selection was involved in the origin of eggs and sperm back in the day, about 1 billion years ago. And it may play a role in sympatric speciation, which we will come to probably in mid-February. So just remember that. Disruptive selection is selection to take a population that has a certain mean value and split it in half and turn it into two different things. Now, natural and sexual selection. We've referred to sexual selection with the guppy. The classic example of sexual selection is the peacock's tail. This is actually what inspired Darwin to come up with the concept. He said, look at that peacock. There isn't any reason from the point of view of survival for a male peacock to be that colorful and have that big a tail and have this absolutely exotic behavior of dancing around waving its tail. And in fact, if you look at the birds of paradise, the amazing thing about the birds of paradise is not really their feathers, it's what they do with their feathers. They do fan dances with their feathers. They can do the rumba, they can shake, they can rock and roll, they do all kinds of stuff. And they're all dangerous because they're out there displaying and predators can come along and eat them, okay? In fact, peacocks are eaten by tigers, or they were eaten by tigers before the tiger just about went extinct in India. They're down to a few hundred in India, and the Siberian tiger is under threat right now in Siberia. But the tigers traditionally ate peacocks. They really did. So the display behavior was dangerous. So what the male was doing was he was trading off survival for mating success. He was a victim of female preferences. Don't tell that to the fraternity guys, okay? So, sexual selection is a component of natural selection. Natural selection is all about variation and reproductive success, and you can achieve reproductive success by mating and by surviving and by doing other things. Okay, so it's a component of natural selection. And the trade-off involved is survival versus mating. It's driven by two things. Either the males are competing with each other for access to females, or the females are conniving against each other for access to males. One of those processes may drive intelligence a little bit more than the other. And it's also driven by members of one sex choosing mates of the other sex. So we're going to have a whole lecture on sexual selection. It's often fun to write a paper on this topic. There are several criteria that one sex might use in choosing a mate. One is a direct benefit. So with birds, that would be, oh, that male's got a really good territory. It's got a lot of food in it. Therefore, I could have a lot of babies and raise them there, so I'll go live in that territory. Not so directly looking at the male, just saying, oh, he happens to hold that territory. That would be a direct benefit. Or you could say, oh my goodness, is he sexy? If I mate with him, my sons are going to be sexy too. That's called the sexy son hypothesis, and actually it does appear to drive some of the more extravagant displays and is probably responsible for the evolutionary shaping of the peacock's tail. A third hypothesis is, oh, he's resistant to disease. And he happens to be wearing a piece of morphology that I can detect externally that tells me that he's resistant to disease because it's expensive to produce and only resistant males are capable developmentally of producing it. There's an interesting principle involved in that. Basically, it is that honest signals are costly. Okay? And if disease resistance is costly and you can advertise your resistance with a signal that you are disease resistant, then that could be something that a female preference might then evolve to notice. We'll go into that, but you can see immediately that if the signal is not costly, then it can be invaded by cheaters. And then as soon as there was cheating going on, the female preference would erode because there wouldn't be any point to having that preference. You were getting cheated on too frequently. Yeah.
I just have a question about the sexy son hypothesis.
Yeah.
It seems, I don't know, it seems like it implies a certain psychology in the mother that's kind of expensive to have.
Yeah, it does, doesn't it? And isn't it interesting that things that we find beautiful evidently are also preferred by female birds and by bees that are locating flowers and things like that? It implies a whole set of innate preferences in choice. It doesn't imply necessarily consciousness. I mean, you can build robots that will do this, but it does imply a fairly costly choice apparatus, which appears to have evolved. Frequency-dependent selection is another kind of selection, and that happens whenever the advantage of doing one thing depends on what the other people in the population are doing. Okay? There are some classical examples of this. One is the classical 50-50 sex ratio, and another is genetic diversity for immune genes. I'll just say a few words about genetic diversity for immune genes because we're going to come back to sex ratios when we do sex allocation theory. Let's suppose that you have a gene that is resistant to a particular disease, and therefore your offspring survive better, and you have more grandchildren, and this gene then spreads through the population until eventually most of the people in the population are resistant. That means that there's selection operating on the disease to come up with a variant that can overcome that resistance. And when that variant comes up, it will spread until it is common, and it creates selection to cause the same thing going on in the host population, and back and forth it goes. The more frequent something becomes, the more it's subject to very strong negative selection, and the less frequent it becomes, the more it's protected from being selected. Because things that are rare aren't very good resources. Things that are common are great resources. And so what happens is that you get what is now recognized as a classical oscillation of virulence and resistance between the host and the pathogen. One of the most interesting things about the human immune system is that the MHC or HLA genes that mediate this kind of resistance against pathogens have some of the highest genetic diversity of any genes anywhere. That looks like variants, rare variants have been selected again and again and again. So every time something becomes frequent, it becomes useless and another rare one is selected. And eventually a huge supply of variation builds up in the population. So this principle really has had quite a role to play in the selection of the vertebrate immune system. Okay, group selection and species selection. I'll go through this fairly quickly. We're going to come back to this issue when we do behavior in April, okay? But group selection is, here's an example of group selection. A bunch of partridges get together in Scotland in late fall. They look around. They notice that there are just a tremendous number of partridges in Scotland in late fall. And they think, you know, speaking anthropomorphically, they think, oh, there are too many partridges. Therefore, we will all cut back on our reproduction so that our population does not go extinct. That's an example of group selection. It won't work because over in the corner is sitting Joe Partridge, who looks at all of these guys and says, you're idiots, you're cutting back on your reproduction, I'm going to have 50 babies. Group selection is vulnerable to selfish mutations. Selfish mutants invade. Okay? So, and they invade for a variety of reasons, and we'll work through all of that. But group selection is not stable. Selfish mutants invade. They do so. There's a lot of selective events in genes and individuals for each selective event of a whole group. And if you extend group selection up to species, how many times have you guys ever heard, perhaps on Discovery Channel or BBC or National Geographic, that behavior X or morphology Y exists for the good of the species? Have you ever heard that? Yeah, happens a lot. It's bullshit. Just plain bullshit, okay? Things don't exist for the good of the species. Things exist because individuals outperformed other individuals in the competition for reproductive success. Now, there is some large-scale differential species selection that occurs on the phylogenetic tree. And it shapes patterns at a big macro scale across the tree. One of them is sex. Virtually all asexual things are relatively young, and they had sexual ancestors. It appears that sex reduces the probability of extinction, and that asex makes you more extinction prone. So that is a kind of species selection. But it's not a selection for a precise adaptation. There's no way that species selection could have ever designed the vertebrate eye, the vertebrate brain, any of the detailed, precise, complicated mechanisms that we know of in biology. All that stuff has gone on because of individual and gene selection. Some of the big macroevolutionary patterns have been generated by a kind of species selection. For example, the fact that the dinosaurs aren't here anymore and the mammals dominate the Earth is a kind of selection. It doesn't tell you about how fast the mammals run, why they are warm-blooded, da-da-da-da-da. Dinosaurs were warm-blooded too. Okay. We can classify selection a number of different ways. Each of the methods of classification highlights a distinction. Selection can be strong and the response can be fast, but some traits evolve very, very slowly. And you need to be able to hold those two facts in your mind and have intellectual tools that will allow you to deal with both situations. Okay, next time, neutral and maladaptive evolution. Leave that off the board, but I do want you to have it so it makes it easy for you to review this when you download it and you look at it in your notes, because it does summarize the main points. Basically, what I'm going to do is tell you that evolution can be either adaptive, in which case it has been driven by and shaped by natural selection. It can be neutral, in which case it's been dominated by drift, or it can be maladaptive. So evolution does not only produce things that work well. Evolution produces things in which stuff can go wrong. And sometimes evolution just wanders around. Now, adaptive evolution is not about the survival of the fittest. That is a phrase invented by Herbert Spencer in the 19th century that has had a long shelf life. So, here's incredibly fast. Antibiotic resistance. It is a curious and striking cultural fact that in the United States, when people talk about antibiotic resistance in television and in the newspapers, they almost never mention the word evolution. They say it emerges or it develops. But in fact, this is the poster child for rapid evolution. If you can see here, roughly the years in which antibiotic resistance evolved in these diseases. If we develop a new drug and release it in the UK in 2009, resistant strains of bacteria will have evolved and will be in hospitals in the UK within 6 months. Today we're going to talk about adaptive evolution, and that means that today is going to be all about the different kinds of natural selection that there are. It's going to be about the vocabulary that evolutionary biologists use to describe selection. It's going to be about rates of evolution, why evolution is sometimes very fast, sometimes very slow. And it's going to be about the different contexts in which selection occurs. So we'll talk a little bit about sexual selection, we'll talk a little bit about group and species selection, things like that. All of these things that I mentioned today are going to be coming up again and again. So this is just part of the intellectual toolkit for dealing with the course. This is an outline of the lecture, and since it's a whole lecture, it's in pretty small type, and I don't expect you to reach wrong. Adaptive evolution is about design for reproductive success. It's all about how many children and grandchildren you have and whether you do it better than somebody else that's in the population. It's always relative. Natural selection is like the tale about the Buddhist monk and the disciple who were attacked by the tiger. And the disciple says to his master, "Oh, master, we're going to be killed because we cannot possibly outrun the tiger." And the master says, "No, I just have to run faster than you." Well, selection is always relative. Okay, it always depends on what the picture is of reproductive success in that population at the time that it's happening. Now, I'm then going to discuss when selection is strong and when it can be slow, and I will tell you something about the rates, the units in which evolutionary rates are measured, and then I'll run through types of selection. Now, there are going to be two questions that pose puzzles that come up in this lecture. One is going to be, what will happen if directional selection continues for a long time? Can that continue? And if it has to stop, then why should it stop? And the other question will be, how can we explain that even though evolution can really be extremely fast, that sometimes things don't change for hundreds of millions of years? So you have to be able to come up with enough intellectual tools to be able to handle that range of variation in evolutionary outcome. Both of those things really do happen. Nothing for a long period of time or incredibly fast.